All right. Hello, everyone, and welcome to Viruses. So, the next, let's say, series of lectures, which is going to be around 3 or 4, we're going to be talking about all different types of viruses, which I think is going to be super cool. We're going to be introducing you guys to all these viruses that are, let's say, making our life in a bit in hell. So, today, with great excitement, I'm going to be talking about DNA viruses. So DNA viruses, so they represent one of the most diverse groups of viruses that there is. And by DNA viruses, we technically mean that the genetic material is made of DNA. So you can really put them into these three families based on actually, their shapes. If they're icosahedral, you have two different types. You have the naked ones, which are more the adenoviridae. So these are the adenoviruses. If they're enveloped, they're more the herpes viruses. And if they're, helical, complex kind of shape, these are more the poxviridae, basically family. And today we're going to talk a lot about the herpes viridae and a bit about also the poxviridae families, because these are the ones that do a lot of basically, let's say, damage or let's say diseases. Adenoviruses, I'm not going to talk too much about them. It's mostly these two that are, let's say, quite damaging to our health. And the thing about DNA viruses, the most medically important ones, they have a primary manifestation with skin lesions. They have a thing with basically creating skin lesions. These, for example, can be, herpes viruses, the herpes simplex viruses, or the varicella zoster viruses, which produce vesicles. You have pox viruses that create these kind of firm, fleshy papules. Then you have the human papillomas that create these papillomas or warts that are really annoying. So you see that there are these kind of herpes viruses that all they do, or even poxviridae family, that are really creating these kind of, let's say, kind of skin manifestations or lesions. So those are very, let's say, easy to notice. Very nice also in the clinic to diagnose them. When it comes more to other DNA viruses like cytomegalo or Epstein-Barr viruses, these ones, they do not produce much of symptoms. I call them more the silent killers. These guys, they infect you, they might do a bit of symptoms, but that's it. They can stay there for your lifetime and then suddenly come up again and do a lot of damage. So that's why I call them the silent killers. So I'm going to be talking a lot about these different types of viruses today. So let's just first of all talk in general about DNA viruses. So as we know, these viruses, as we said, they all share basically the same genetic material, which is DNA. But there's small little, let's say, differences. This could be in their organization and replication strategies. With their basically their organization, it all depends on, how is the DNA? Is it basically linear or is it circular? Because we see that herpes viruses and pox viruses all have linear double-stranded DNA. But human papilloma viruses is a bit different. The papilloma viruses, basically, they have it much more, let's say, double-stranded circular. So, bit different in this kind of sense. So again, herpes viruses, the pox viruses are this linear stranded, while the papilloma viruses are more, let's say, circular. So that makes them different in their organization. But of course, as we know, DNA viruses, majority of them or almost all of them, what they're going to do is replicate in the nucleus. Because they have DNA, they require certain machinery which are found in the nucleus. They utilize actually the machinery coming from the basically host, which is found in the nucleus. There's only one exception to this, which is the pox viruses. The pox viruses is the only DNA virus that can replicate in the cytoplasm. Why? Because it does not need host transcriptional machinery. It has its own ones, so it doesn't need to go into the nucleus to actually, be able to transcribe and do all of its genetic material synthesis. So pox viruses is the only exception towards that. So when it comes to viruses, I'm really gonna dive a lot today into herpes viruses, and then of course I'll be talking about other ones specifically. I'll be talking about in general the pox viruses. So when it comes to herpes viruses, it's a family that includes several different types of clinically important human pathogens. These could be your herpes viruses, the herpes simplex virus. I'm quite sure you've heard of them. These are the herpes simplex virus type 1 and type 2. This is also the varicella zoster virus, cytomegaloviruses, Epstein viruses, and also the human herpes virus 8, which we call also Kaposi sarcoma associated virus. So these viruses, what they share is they have a very similar structure and biological features, but they all basically have a linear double-stranded DNA genome. And all of these herpes viruses, what they also share is they can also do a lifelong latent infection, meaning they can insert their genome into ours and they can basically stay there for a lifetime, hence lifelong latent infection. So they can technically go inside, infect us, create a lot of symptoms, inject their genome in our genome, and then suddenly no symptoms. They enter this latent kind of phase where there's no DNA, there's no viruses being produced, there's no viral proteins being produced or replicated or anything. They enter this latent phase and they can sleep. They can sleep for months, for years, for literally decades even. So this is where we say this lifelong latent infection. So it's very, quite important to know because, of course, what happens is during this latency, we might not have any symptoms, but when they get reactivated and they start to replicate again, they start producing a lot of symptoms. So clinically, it's very important to distinguish these kind of different types of phases and when to basically start, treating people. So the structure of the herpes viruses in general, they're quite big. They're about 120 to 200 nanometers in diameter. It's actually among one of the largest viruses, pox being basically I think it's actually the biggest virus, then followed actually by your herpes viruses. So generally basically DNA viruses are quite big in general. So 200 nanometers is quite generally big for a virus. The herpes viruses, they have this icosahedron basically capsid. So basically they have this really nice kind of icosahedron shape. But what they also have is an envelope. So this envelope is usually taken up from the nucleus. So this membrane, they actually take it from the nucleus because the herpes viruses, they infect basically and they go to the nucleus, and inside the nucleus, once they produce now basically your virion, what's going to happen, this virion is going to literally snatch basically this membrane from the nucleus. So it's going to wrap itself with nuclear membrane. But between basically the envelope, which is shown here in E, and the capsid shown in C, you have a lot of these proteins that are called tegment proteins. These tegment proteins are extremely important when it comes to viral replication and basically regulatory proteins. So they really help in regulating certain, let's say, machineries or certain, let's say, replication of your DNA and also viral replication. So literally between the envelope and the capsid, you have a bunch of these tegment proteins. And we'll definitely talk a bit about a bit of their role, what those are doing. So one thing about basically, as we know, as I just said, they love these herpes viruses. They love when they infect to go to the nucleus because that's where they basically start to replicate their genome. They use a lot the host machinery to do that. And then basically once they're all done, they're able to, let's say, kind of steal a bit of the nuclear membrane to create this envelope for themselves. But one thing that we also see that is very characteristic of herpes viruses is they love to really create these, let's say, inclusion bodies. These inclusion bodies are literally, again, as we said, these kind of focal areas where you see a lot of viral replication happening. So we can see basically microscopically, if we take an infected, let's say, kind of skin graft or whatever, basically kind of, let's say, kind of lesion, you basically scrape a bit of a lesion, and you see these basically these cells with these kind of very characteristic bodies that we call inclusion bodies. They can already tell you, hey, you're most likely infected by herpes viruses because they all produce this kind of stuff in that kind of sense. And one thing also that these viruses love to do is latency. So something that we spoke about, once basically they infect, they will literally insert their genome into our genome. They stay there and they basically go into this latency, meaning that they're not producing nothing. Nothing is happening, no viral replication, and they're just there. No symptoms. Suddenly some sort of stress or stimuli is going to cause them to suddenly now start to reactivate and start to literally start, producing much more virus and then causing us all these symptoms. And of course, this latency is lifelong. It stays there for the rest of your life because the DNA is inserted, the viral DNA is inserted into our own genome, so into our chromosomes per se. The mechanism of latency is not really that well known. for many viruses. But for HSV, we've known to started to really do a lot of studying for this. So what the herpes simplex viruses, which have denoted as HSV, they produce something called a latency associated transcript. So it's a transcript, it's an mRNA transcript that produces a lot of microRNAs. So these microRNAs, their job is to shut down viral replication. So it stops viruses, specifically the herpes simplex viruses, from replicating. So that's why in the latency phase, they do not replicate because of these LATs. Cytomegaloviruses also use very similar thing where they use also microRNAs. These microRNAs, they inhibit any viral mRNA being needed for replication. So they're just literally suppressing any basic protein translation. So that's why it stops viral replication in general. CMV has extra stuff. What it does, it also stops basically the cell from going into apoptosis. Because if the cell dies, the CMV basically genome also goes with it. So it kind of prevents it from dying. So it's able to survive and keep its genome staying there. So that's why it can stay in its latency form for quite a long time. So the reactivation of these latent herpes viruses, these can be coming from certain stimulus. We don't know for many of them. Some of them, for example, we know for herpes simplex viruses, this can be sunlight exposure, this could be fever, this could be stress, this could be trauma. Really the full mechanism of how these certain, let's say, stimuli reactivates this virus is very unknown. It's actually, we don't know much about this actually. But what we know is that the reactivation, so once you're infected, you have symptoms, then these viruses, they go into latency, so they suddenly stop, no symptoms. The reactivation of them causes again to bring up these, let's say, symptoms back again. So it's very important clinically to see, the recurrence and transmission of these kind of viruses in general. So when it comes to the classification of herpes viruses, so herpes viruses is a family. You have basically 8 different types. You can call them basically herpes, the human herpes 1, 2, 3, 5, 6, 7, and 8, on and on and on, basically. But you can also put them in subfamilies. And I don't want you to know the name because, to the point, this is very medical. I don't even knew that there was even names until my brother, who's a doctor, told me about them. I knew more the subfamilies and their common names. So I want you guys to know the subfamily and also the name. So we have the alpha family. The alpha herpes viruses are the HSV1, the herpes simplex virus 1 and 2, and also the varicella zoster viruses. We call them alpha purely because they love to actually infect epithelial cells and establish latency in neurons. So that's why we call them the alpha viruses, because they love to establish the latency in the neurons. Then you have basically the beta herpes viruses. These are cytomegaloviruses. Cytomegaloviruses are quite weird in a sense. They will bind, or they will literally kind of, let's say, infect all different types of cells. So it's a wide range of tissues that they can actually kind of, let's say, do or infect. Then you have the gamma herpes viruses. The gamma are the Epstein viruses and the human herpes virus 8, which we also call the Kaposi sarcoma association virus. These ones, they primarily infect lymphoid cells. This could be your B cells, this could be your T cells, or these could be your NK cells. So they have a preference. The gamma herpes viruses, they love to go more for the lymphoid, and they're more associated with malignancies. They love to induce cancer. So the classification helps to explain, and generally, the differences in the disease patterns and the tissue tropism. So that's why look at the family. So as I just, kind of briefly said, these herpes viruses, what they love to do is they do also can induce cancer. Specifically, basically the gamma herpes viruses, like your Epstein-Barr viruses, they can produce basically Burkitt's lymphoma, which is a lymphoma. It's a B-cell malignancy. Epstein-Barr viruses, they love to actually infect B cells. And what they can do is they can mess up a lot with their proliferation and apoptotic signaling. So they can really mess it up. So they can stop their apoptotic signaling and they can induce a lot of proliferation signaling. So it kind of makes the B cells start to divide uncontrollably. But because the Epstein-Barr viruses, they can also in some way bind to epithelial cells, they can also cause the same thing, but in epithelial kind of context. For example, in the nasopharyngeal carcinoma. You have another one, basically the human herpes 8 virus can cause Kaposi sarcoma, so a different type of cancer as well. So again, they do this by really messing up the cell proliferation and apoptosis machinery or signaling within a cell. But of course, these herpes viruses, other than doing it on basically just humans, they can do it on many different types of animals, showing how broad their oncogenic potential is with these viruses. So now I've spoken in general about these viruses. Let's go into the details of most of these viruses because they're quite interesting. So now we have the most common types of herpes viruses, which is your herpes simplex viruses 1 and 2. Quite sure that you definitely have heard about them. The herpes simplex 1 and 2 structurally and genetically are extremely very similar. It's almost impossible really to know the difference when you look at them under a microscope. They differ very in the angiogenicity. So meaning that their literally surface proteins, they have different, let's say, sequences. That's the smallest difference that they really have between the HSV1 and 2. So these small little differences can actually has a certain tropism, can change their tropism a bit. So what we see clearly is with HSV1, it's mostly you find them in your oral and facial regions. They really produce a lot of infections above the waist that are mostly in your oral and facial regions. HSV2, they produce much more, let's say, kind of infections below the waist, specifically your genital area. So if you see certain, let's say, kind of infections, and it's a herpes viruses, usually if it's above the waist, type 1, so the herpes simplex 1, if it's below, herpes simplex 2. But this is not a, let's say, basically a rule that it's basically 100% true because of human behavior, particularly oral and genital contacts that we've been having among humans. We started to see a lot of herpes simplex virus 2 in the mouth because, of course, your genitals can be infected with HSV2, and with oral contact to the genitals, you can actually transmit the HSV2 to basically the mouth. So that's where you see differences. This is all because of the human behavior. You also can see HSV1 being transmitted to your genitals. So it's not basically a strict rule that if the infection is above the waist, it's HSV1 only. No, that's not true. So as I said again, the differences, structurally when it comes and biologically to HSVs, to the 1 and 2, structurally they're you cannot really distinguish them under a microscope. They're very similar, have an icosahedral capsid, both have an enveloped virion, both linear double-stranded DNA. The difference that I'm saying is mostly on their G protein. Their G protein is this basically protein that binds technically to the host receptor, and there's small differences in the amino acids there. That's really what makes them different in that kind of sense. So really, it's quite really hard to actually, let's imagine that you have, let's say some infection in the mouth, it's a herpes virus, you look at the microscope, it's very hard for the microscope to see if it's 1 or 2. So what you would do is basically use specific antibodies that are able to distinguish them, or you use them PCR to actually really say, hey, is this type 1 or type 2? So the initial steps of the herpes simplex infection, what happens is that in specifically the proteins of the herpes simplex viruses, specifically the G protein, the glycoprotein, what it does, it binds first to this heparan sulfate molecule. This heparan sulfate molecule is found basically quite extensively, specifically in your epithelial cells. And this basically allows the virus to kind of sit on top of the cell. And this then allows the virus to bind more specifically towards nectin. So it's other glycoproteins, it's going to bind also to nectin. And nectin is what's going to really push it to actually create this, let's say, host membrane fusion. So it helps the viral envelope fuse with the host cell membrane. So it always starts with heparin binding. This then allows, again, the binding of nectin. When you have the binding of nectin, this really pushes the fusion of the membranes together. Once basically you have the fusion, the capsid is released. Along with the capsid, you have the tegmon proteins. Remember, so you release, basically the envelope is basically now fused with the membrane, basically. you have the tech-bound proteins along with the capsid. So what happens with basically is that they literally are, transported. So they're taken up. They specifically in your neurons. So in the epithelia, they're taken up and they go straight to the nucleus. In the neurons, what happens is technically they are, taken up, and they're basically using some sort of these kind of microtubule motor proteins, specifically dynein. They're literally going to transport them all the way from these little dendrites all the way to the body of the neuron, and where once they reach the nucleus, what's going to happen technically here is that the virus is going to release its genome, specifically its linear double-stranded DNA. This linear double-stranded DNA is going to be able then to go inside. It's literally going to go inside the nucleus along with the tech-bound proteins. Specifically, VP16 is one basically tech-bound protein that really goes inside the nucleus. So once inside the nucleus, this is where the tech-bound proteins are super important, because what the tech-bound proteins are going to do, they are going to start the activation or literally the production of the immediate early genes. So this VP16, this tech-bound protein, is going to induce now the transcription of early immediate genes. So that's why we need tech-bound proteins, because this immediate early phase technically relies on not having already basically viral protein synthesis. It relies that there's already a protein there, so which is this VP16. That's why with these herpes viruses, you need tech-bound proteins. They really start off these replications. So once basically you have VP16, what it does, it basically starts to induce these immediate early genes, and you start to getting them, the transcription of them and translation, and you have basically 5 regulatory proteins being produced. These 5 proteins, what they're going to do is going to kickstart also the transcription and translation of your early genes. What are these early genes? These are your DNA synthesis, your nucleotide metabolism proteins. All of these ones, these are basically needed for viral DNA replication, but also these are like DNA polymerase and thymidine kinase. All these different ones that are going to help, duplicate the genome and do a lot of this kind of viral machinery for replication. And then these basically are going to kickstart the late genes. These are more the structural components. These could be, for example, basically all the stuff needed to assemble the capsid. So anything that needed for the viral. So not all of the basically the DNA of the herpes simplex viruses are literally expressed at the same time. No, they come in phases. You have these immediate early genes, which are basically kick-started by VP16. These immediate early genes then kick-start your early genes, which then kick-start your late genes later on. And this comes basically in packages. So once basically everything is packaged, you have DNA replication. Basically now the genome is duplicated. You have all the structural proteins creating the capsid. Beautiful. The capsid now what it does, it literally does this literally nuclear budding. So where it's literally going to bud off and steal a bit of this membrane from the nucleus, and then it's literally gonna go out. And once it goes out, by the way, this is not the best picture, because this picture you see the nuclear capsid, leaving it by its own. Usually, no, it's still surrounded by a membrane. So what happens that when it actually moves outside, usually this budding off does not kill the cell. So this is very important because it actually keeps these cells still alive in some way to keep basically this latent infection. So it's quite important in that kind of sense. So one thing is that, of course, usually when we are, let's say, infected by herpes simplex viruses, they infect us first basically in our epithelial. So in the mouth, for example, or in our genital area, specifically epithelial cells. What these viruses can do is they can actually move on and they can start infecting neurons, and specifically sensory neurons. And where they establish latency. This is where they're going to insert their genome specifically there. So what happens, basically, let's imagine now I have HSV1. So basically I managed to get infected through my mouth. Basically the herpes simplex virus does all the infection in my mouth. Then it goes into the neurons, specifically these sensory neurons. What happens with HSV1? It's literally going to go into my neuron. It's gonna be, same thing. It's gonna bind to basically this heparan sulfate, this nectin, and then it's gonna release its capsid. The capsid is literally, along with the tech-bound proteins like VP16, is literally going to be transported from literally the axon terminals all the way towards the cell body and nucleus. And basically it's all because of these macrotubules. And we call this movement, we call this retrograde transport, where it's a transport going from your axon towards your cell body. So it's literally gonna go there, and once there, it's gonna go into the nucleus and literally insert its genome there. And it's gonna be reliably, it's become dormant. Basically, that's where it's gonna stay. HSV1, usually it likes to go into trigeminal ganglias, while with HSV2, it likes more lumbar and sacral ganglias. So again, these small little differences in the G proteins kind of gives them different tropisms, so different places where they like to infect or create latency. So once they're there, basically they insert their genome and they're latent. They do not do anything. Some sort of stimulus happens, something occurs that the genome, the whole virus wakes up, starts producing basically virions, all these different types of viruses, and again they start to move in the opposite direction. So from the cell body towards the axon terminal, we call this the antegrade basically transport, so the opposite. So, and then they basically are released. They then start basically to infect your epithelial cells and creating the symptoms. So basically this is exactly what I was saying here. What are these symptoms? Well, basically these are the most annoying symptoms, which are technically you have the formation of vesicular lesions. Basically are either on the skin or the mucous membrane. So this could be, for example, in your mouth, or this can be all around your genital areas, be it the anus, be it basically the penile area, or being also the vulva or around the vaginal membrane. So all of that. So as I said, HSV1 is mostly found in your oral labial infections, so more in your mouth. HSV2 is usually associated more with genitals. But again, usually they're capable of infecting either location. Of course, these HSV can other than just localized lesions, in general, they are localized, they stay there. But in some cases, when specifically people that do not have a good immune system, they can be systemic because we have epithelial cells all over. So they can really go all over the place, particularly in immunocompromised or neonates, so babies. So they can disseminate and affect multiple organs. But if you're a healthy individual that has a healthy, intact immune system, they're just localized lesions. And the thing about the skin lesions, they're very particular. They have this very characteristic sequence that is very important for recognizing it in a clinic. So what usually happens at the beginning is you have localized cell destruction and inflammation. So you create this bed, this really big bed of redness. So this is called erythematosis. Forget these basically vesicles. Erythematosis is basically this over here that I'm pointing out with my mouse. It's this bed of redness. That first comes up. Then you get the vesicles formation. So these vesicles are filled with fluids, and this fluid is extremely rich in virus. So these are extremely contagious. Then these basically fluid sacs will basically pop and will basically start to open up, and they literally start to rupture, and they start to bit by bit ulcerate, and they basically start to, in some way literally crust over and heal. So this progression of vesicles, basically first of all this erythematosis bed, vesicle formation, popping of these basically vesicles, crust formation. This is very clinical HSV, so herpes simplex viruses. You see this is basically a clinician will already say you have herpes viruses. It's as simple as that. So usually the primary herpes simplex infection, this basically means it's the first time you ever get infected with these viruses. You do not have a pre-existing immunity. You don't have memory B cells or memory T cells to basically kind of combat it. And usually the first time it's the most severe. You have a lot of basically these vesicles happening because you don't have an immune system that is able to stop it. So it can take longer. It can produce fever, and the healing time can be prolonged. But then basically once it goes into latency and then gets reactivated, the secondary, let's say, infection in your epithelial cells, it's much more milder because now you have a B cell and T cells that are controlling it and basically being able to kick it out. So usually the first infection is much more, you have quite a lot more of these basically vesicles compared to your secondary. Or your basically, not your secondary also, but your reactivation of the virus. So again, as we said, HSV1 is more oral labial disease. It mostly happens basically in your mouth. Again, the HSV2 is more the genital areas, as I was just basically saying. But again, these are not, it's not basically a must. HSV2 can also be present in the oral labial. The most, let's say, dangerous thing that can happen, per se, is neonatal herpes. Neonatal herpes is basically when when you get herpes as a kid. Usually this happens when you have an infected person kissing a baby. You kiss them in the mouth, or you kiss them around in some way or the other, mostly in the mouth. It's basically through that. You can basically cause babies to actually get infected with herpes simplex viruses. This is why it's strongly recommended not to kiss your baby in the mouth. Because if you do have herpes and it basically goes to the baby, it can kill it. Because the baby does not have an intact or proper immune system. So what happens is that the HSV doesn't become localized. It starts to disseminate to many different types of basically organs. You start to have eye and mouth diseases. Basically, it can go into your central nervous system. And specifically, what can actually do is encephalitis. It can really go into your brain and cause a lot of basically damage in your temporal lobes of your brain. This can cause fever, headaches, confusion, seizures, and altered consciousness. So it's very important that you do not try to infect babies with herpes. And usually the type of herpes that you get infected with neonates is HSV1. And that's why we usually say HSV1 is causing basically encephalitis. So the immune response to HSV, we do get both the innate and your adaptive immune responses being there to control it. But mostly what's super important is your cytotoxic T cells. Because these ones are the ones that are keeping them basically from, spreading too much. So, and generally, as we said at the beginning, you have no immune response, so the virus is able to do much more damage through your secondary exposure or your reactivation. Your T cells come in and they really kind of stop them from doing much more damage. So, the HSV is extremely good at evading your immune system, so it's able to snatch away the MHC1 molecules, so you avoid your cytotoxic T cells from killing them, but they can also stop basically specifically certain cells from dying. So they keep them alive so they can keep producing more and more and more viruses. But also them being in a neuron is already evading the immune system, because your immune system doesn't want to kill neurons because we cannot recreate them. So them being already there is very important. But also having this latency in the neurons, so having the genome inside there keeps them for life. So that's one way of also evading your immune system. So transmission, as we know, HSV is usually transmitted through your non-sexual contact during childhood. This could be through kisses. Basically, it's non-sexual. HSV2 is mostly done through sexual activity. So those are the ones that basically, that's why in general we get to see primarily HSV1 in our mouth and HSV2 basically in our genital areas. The thing about it is that one thing that I also it's a nice thing to know, it's not that if you basically are infected with HSV1 or 2, you're going to get symptoms. Actually, around 20% of people only get symptoms. So by the age of, let's say, 50, 60, I would say a good 80 or 90% of people have herpes viruses. It's only 20% that actually get symptoms. Why? We really don't know that. It's not really understood. But one thing we know is that the prevalence of HSV keeps growing and growing and growing. So it's very important to understand the dynamics of transmission. So, we need to try to prevent it. This most importantly comes with sexual practices. So, by wearing a condom, but also basically childbirth.